The foreign body response to implantable biomedical devices is considered to be primarily a reaction to the chemical composition of implantable materials. In this study, we show for the first time that mechanical signaling is sufficient to drive the biology of FBR independent of material chemistry. I'm Jagan Padmanabhan, a postdoctoral fellow in Dr. Jeff Gertner's lab, and here's how we came to that conclusion. Let me start with some background. Surgical implantation of medical devices initiates a wound healing cascade that evolves over time into the classic foreign body response. The end result is the formation of a thick fibrous capsule, which isolates the device, disrupts its surrounding environment, and leads to eventual implant failure and rejection. Nearly 90% of all implants failures are associated with the FBR. The specific molecular and cellular mechanisms that underlie FBR have not been identified. The lack of this understanding is exacerbated by the inability of standard mouse models to recapitulate the highly fibrotic response associated with implant failure in humans. To elucidate the molecular mechanisms underlying FBR, we went back to the drawing board and started from scratch. We collected breast implant capsules with different degrees of severity of fibrotic encapsulation. Fibrosis around breast implants is classified using the Baker system, where Baker 1 is the least severe and Baker 4 is the most severe. Analysis of the human specimens with a biomarker panel revealed that the genes that are upregulated in Baker 4 implants were heavily involved in mechanical signaling pathways as well as inflammatory signaling. In contrast, Baker 1 implants displayed modest upregulation of glucose and fat metabolism, suggesting normal metabolic activity. Since elevated mechanical stress has been previously linked to the activation of inflammatory signaling, we postulated that upregulation of mechanical signaling pathways might causally drive cell responses toward a more Baker 4-like pathological foreign body response. Next, using finite element modeling, we found that owing to the large differences in tissue geometry and tissue mechanical properties, the effective mechanical stress around biomedical implants in humans was 100-fold higher than a similar silicone implant in mice. To investigate the importance of mechanical signaling in FBR, we developed an implantable device encapsulating a small coin motor, which could be induced to produce intermittent in situ implant vibration. Finite element modeling predicted that these mechanically stimulating implants, or MSIs, would generate a maximal stress of 24.1 kilopascal, which is similar to that surrounding most human implants. Thus, MSIs provide a unique platform to examine the effective mechanical environment alone on the subsequent FBR. And we found that the tissue architecture of the FBR capsule produced by MSIs was very similar to that observed in humans and markedly different from standard neighboring implants. Specifically, MSI capsules had significantly increased collagen deposition, increased collagen maturity, and a higher level of activation of myofibroblasts, which was nearly identical to that of human specimens. Next, we analyzed cells surrounding the murine MSI and standard implants using single-cell RNA-seq. We analyzed a total of 36,000 plus cells. Standard implant cells initially expressed a modest activation of inflammatory markers, such as the IL-1 beta, which subsided over time. In contrast, MSI capsules showed a robust early activation of these markers, which progressed in later time points. This pattern of gene expression was observed for a large number of common inflammatory genes. We identified three major cell types in these implants: the myeloid cells, the lymphoid cells, and the fibroblasts, which were then individually analyzed. Myeloid cells alone clustered in 8 distinct subpopulations, and of these, clusters 1, 4, and 7, which were composed of macrophages and granulocytes, were specifically enriched in the MSIs. Next, we examined how the transcriptional profiles of these murine clusters compared to the expression of the Baker 4 human markers. The combined gene expression of the top 25 Baker 4 biomarkers, called the human Baker 4 gene signature, was highly upregulated in the MSI clusters as compared to the standard murine clusters. Next, we analyzed fusogenic macrophages. Fusion of macrophages leading to the formation of foreign body giant cells is a hallmark of the classic FBR. We found that cluster 4 macrophages that showed a simultaneous upregulation of ARG1, RAC1, and MMP14, a transcriptomic profile that is distinct to fusogenic macrophages, were highly enriched in the MSI capsules. And since macrophage fusion is promoted by activated lymphocytes, we next analyzed those cells. MSI lymphocytes showed a preferential upregulation of MHC class II foreign body response components, which is a well-known promoter of macrophage fusion. And then on to fibroblasts. Fibroblasts in the standard movie model showed upregulation of proteolysis and markers and signaling associated with the negative regulation of cell proliferation, all these indicating a resolving gene phenotype. In contrast, MSI fibroblasts showed the upregulation of myofibroblast markers such as PDGFRA and profibrotic cytokines such as CXCL2, indicative of a more active phenotype. This leads to increased differentiation of myofibroblasts and collagen deposition in MSI capsules, as demonstrated before by immunostaining. So in conclusion, by waiving only mechanical stimulation, we were able to recreate a human-like pathologic FBR at the molecular, cellular, and tissue level in mice. Collectively, these findings provide novel insights into the FBR, which carries significant implications for improving the design, safety, and of all implantable medical devices. And finally, I would like to thank my mentor, Dr. Gertner, and all our collaborators, the Plastic Surgery Foundation for funding, and the organizers for the opportunity to present. Thank you.